Hey guys, welcome back to Physical Biology. I know you guys are having paper 3 exam tomorrow. That's why I'm here to tell you guys the 8 tips to get A in SPM Biology paper 3. Alright, so remember to stay until the end of the video because we have the most important tips in the end of the video. Alright, so let's get started. Tips number 1. Just like playing games, before we play the game, we need to know the rules. If you want to score well in paper 3 biology, we need to know the rules first, plan well, and only we can beat the rest. Alright, so let's get started. So let me tell you what are you going to do exactly on that day. Before the exam start, you need to lapor diri. If the exam is going to start at 8 AM, the exam is going to end 2 hours and 15 minutes later, which is 10:15 AM, which means that you guys are going to complete your physics exam, bio exam, and chemistry exam within 2 hours and 15 minutes. Alright, now let's focus on paper 3, this column. You guys must take note that each of the subject will give you 15 marks. Alright, let's look at the last row. Sesi merancang 5 minutes setiap item, masa menjawab 40 minit setiap item, which means that you are having physics, chemistry, and biology. Each of the subject, you will have 45 minutes to complete the exam. And within these 45 minutes, you are going to spend 5 minutes plan the experiment. So the timing here is very, very important because you need to plan and allocate the time properly before you take the exam. But what do we need to do in the sesi merancang? And what do we need to do when we are in the masa menjawab? Now, no worries, I'm going to show you the answer in the next slide. So within the first 5 minutes, you guys are going to check the apparatus and material on your table by using this checklist. The bigger 50 ml with solution A, B, C, D on the table, then you just need to put a tick to indicate you have the figures. If you have 5 petri dish on your table, then you need to put a tick here. Now remember to test the stopwatch first before you carry out the experiment. If you found out the stopwatch is not working anymore, please raise your hand to ask for help. Remember that, alright? So checking the apparatus and material, you are going to spend about 2 to 3 minutes. Tips number 2. Now once you have checked all the material and apparatus, it's time to read your questions. Reading the question is just not reading the question. We actually have some technique to read the questions. The purpose of you to read the questions is to find out the MV, RV, and if possible, we need to find out the CV as well. So what is MV? MV is the manipulated variable. This is something that you need to change in your experiment. RV is the responding variable. And responding variable is actually the result that you can observe in the experiment. Now let's move on to CV. CV is so-called the constant variable. Now for constant variable is something that you want to fix in the experiment. Normally we will not able to find the CV in the questions, but you definitely can find the MV and RV in the questions. Let's do some practice to find out the MV and also the RV. So let's look at this example here. You have to carry out an experiment to study the effect of the concentrations of albumin's suspension on the rate of the pepsin. Now what is albumin? Normally albumins that we prepare by your teacher is actually the egg white. So if you have no idea what is albumin, better go and find it out. Now let's come back to the questions. Look at this. In this experiment, you want to study the effect of concentrations of albumin suspension, which means that you are going to change the concentration inside this experiment. For example, you are going to use the high concentrations of albumins for experiment A, and then you are going to use another concentration with lower concentrations of albumin suspension in the experiment B. So the things that you change in the experiment is called the MV. So every time when you are reading the question, remember to put a bracket or underline the MV. Put a MV above the sentence. Continue reading. on the rate of reactions of pepsin. Rate of reactions will be different when you change the concentration of albumin suspension, which means that the rate of reactions of pepsin, this will be the RV. It's the result that you want to get in your experiment. Alright, so write the RV below the sentence. Done. So this is the first step you guys need to do. Find the MV and the RV. Alright, so let's try the second questions here. You have to carry out an experiment to show that amylase hydrolyzed starch to maltose. This is the case when you cannot find the MV. MV is missing in the questions. So what you need to do is you need to find it from your procedure. So from this question, you can only find the RV, which is how fast the amylase can hydrolyze the starch into the maltose. If you go and study the whole procedure, you will understand that in this experiment, we are actually trying to use different temperature to find out how it affects the rate of hydrolysis starch to maltose. So you must be smart to find out the MV and RV. Sometimes it's inside the procedure, but most of the time, MV and RV can be found in the question. Alright, tips number 3. You guys need to sketch the procedure. One picture speaks 1,000 words. When you come to planning the experiment, it's a bit tough for you to just read the sentence and then imagine the whole process in your brain. So the things that you need to do is, every time when you read the procedure, you need to sketch it out. Now you might think that this is kind of waste of time. No, it's not. Sketching a diagram can help you to clear your mind. It can actually help you to plan your experiment in a more systematic way. Where are you going to draw or sketch the diagram? No worries, your teachers are going to provide you an extra paper sheet. So on this piece of extra paper, you can use your pencil to draw and convert the whole procedure into a diagram. Let's try to work on it. So in this question, you can see, label 2 boiling tubes X and Y as shown in the diagram 1. We are going to label it. It is important for you to do this so that you will not confuse. Now let's look at the second step. Pour 3 ml of 1% of starch suspension into the boiling tube X and Y using a 10 cm cube of Michelin cylinder. So you are going to use a Michelin cylinder and measure 3 ml of 1% of starch suspensions. This suspension is going to pour into X and Y boiling tube. Once you have the starch suspensions in your boiling tube already, you are going to prepare a water bath at temperature 37 degrees Celsius in a 500 cm cube beaker with a volume of 400 ml. So you are going to use a beaker, 4 ml of water, and the temperature must be 37 degrees Celsius. How do you know it is 37 degrees Celsius? You are going to use a thermometer to measure the temperature. So this is for your first set of experiment. Now B, you are going to prepare another water bath at temperature 100 degrees Celsius. Okay, so this time you are going to use a beaker as well with 400 ml of water inside, and then the temperature must be 100 degrees Celsius. How do we know 100 degrees Celsius? You are going to use a thermometer. How are you getting 100 degrees Celsius? You need to boil the water. That's how you prepare this experiment. Yeah, of course, the procedure is more than that. From here, I just want to show you, drawing is the most important things to do before you carry out your experiment. Alright, so guys, let's move on to tips number 4. For tips number 4 onward, we are going to focus on answering technique. Now, observation question is very simple because anything that can use your 5 senses to detect, that one will be your observation. Now remember, rate of reaction is not the observation, but the bubbles that comes out, the things that you can see, the things that you can smell, the things that you can touch, that one is your observation. And of course, during the experiment, we seldom use our taste. Most of the time, we will use our vision and hearing. For example, you can see the color change. For example, you can hear something like the pop sound that it created when the hydrogen gas explodes, and so on. So this is all the observation that you need to put in the answer. So let's take a look for the first questions. Carry out the experiments. State your observation in the boiling tube Z. Okay guys, since we didn't have the whole experiment here, I will just briefly tell you what we were doing in the experiment. Alright, so in this experiment, for this question, we actually have 3 boiling tubes. Boiling tubes X, boiling tube Y, and boiling tube Z. X, Y, Z. In these 3 boiling tubes, we will have different concentrations of the albumins. For the first boiling tube, we are having 5% of albumins concentrations. Y, we have 10% of albumin concentration, and Z, we will have 15% of albumin concentration. In each of the boiling tubes, we are adding in the pepsin. Now, if you guys still remember, pepsin is actually an enzyme. This enzyme will digest the protein into the polypeptide. So in this experiment, we are actually trying to find out how fast the protein is being digested into the polypeptide. Now, if we are doing the experiment, you will notice that at first, the albumin solution, it looks a bit cloudy. And once the albumin is being digested by pepsin to a polypeptide, these albumins will convert into polypeptide, which look more clear. So you are not going to know when is the protein changing into the polypeptide, but you actually can see the cloudy albumin, it turned into a clear solution. So cloudy to clear is actually something that you can see. So this is the observation. This observation will be cloudy turned into clear solution. But then this question is specifically mentioned boiling tube Z. That means you need to write about boiling tube Z, not Y, not X, but boiling tube Z. So in the end, the answer for this question will be like the mixture in boiling tube Z turns from cloudy to clear in 10 minutes. So this is the result that we got in the experiment. If you are doing the experiment, you should know the times for it to turn clear. Alright, so what you can see here, we actually have something that we can see. And this observation can be made in the boiling tube Z. That's all. You can get 1 mark here. Alright, so let's try another question. Carry out the experiment, state your observation in the test tube which contains the starch amylase mixture tested with Benedict solution. Let me briefly tell you what is this experiment about. This experiment is going to use the starch amylase mixture. And we all know that amylase is the enzyme which will convert the starch into maltose. And we all know that maltose is the reducing sugar that will form the break red precipitate in the Benedict solution. So break red precipitate will be the observation that we can make by using our eyes. So from here, we are going to write the answer like this. The mixture form the brick red precipitate. Alright, so that's all for the observation. Now let's move on to tip 5. In tip 5, I'm going to show you how to write a proper hypothesis, relationship, and conclusion. This technique can apply in these 3 types of questions. And I know hypothesis, relationship, and conclusion quite simple to do that. There's one thing that you guys must take note, which is MV always start first, then only you continue with RV. That's why at the first part of the video, I said it's very important to find out the MV and the RV first. Okay, so if you don't know what is the MV and RV, you are not going to be able to conduct the experiment. Alright, so let's get started. Now look at this question. Explain the relationship between the rate of reactions of pepsin and the concentration of albumins. Now, first of all, we need to find out which one is the MV and which one is the RV first. This question is tricky because they put the RV in front of the question. Rate of reaction of pepsin should be the RV. Concentration of albumins is the things that we want to test out. It is the MV. We want to put the MV first, then only we write the RV. So the answer that you should write in this question is the higher the higher. We use the higher and higher technique. So we need to write the higher the concentrations of albumin, the higher the rate of reactions of pepsin. And of course, this question do ask you to explain the relationship. So every time when you write the relationship already, you need explanation behind the answer. So right here, you are going to write down the explanation, the theory part lah. For the theory part, I'm not going to show you here because I just want you to focus on the technique to answer the relationship questions. Let's try another relationship questions here. Based on your diagram graph in C, state the relationship. You can see there's a relationship here between the concentration of sucrose solution and percentage change in the mass of the apple strip. Find the MV and RV first. Concentration of sucrose solution will be the MV. Percentage of change in mass of the apple strip will be the RV. So you can write the answer like this. The higher the concentration of sucrose solution, the higher the percentage change in the mass of the apple strip. That's it, simple only. Alright, so this one no need to write any explanation because the question asks you to state, no need to write explanation. And this question only give you 1 mark. Okay, alright, so tip number 6. Operation definition. This is the nightmare for all the students here. Most of the students, they don't know how to write the operation definition. But operation definition is actually a very simple question if you know the formula. For operation definition, you can see this question normally 3 marks are provided to this kind of questions. That means you need to write down at least 3 points for these questions. The first point that you need to write down is the definition of the object that's stated in the questions. What is the object here? Now let's look at these questions. State the operation definition for the hydrolysis of starch by amylase enzyme. This will be the object. We want to write down the definition of this object. The second point is you want to write down how could you get the definitions of the object by doing the experiment. So normally you will include some magic words, for example like determined by, measured by, or shown by. Okay, now the third one is determined by, measured by, shown by. Shown by what? It is shown by the RV, the thing that you can observe in the experiment to give you the definition of the hydrolysis of starch by amylase enzyme. Now the last one should be how this object is being affected by the manipulated variable. So in the sentence, you are going to write down the magic words affected by, and you write down the MV here. Alright, so theory is just the theory. Let's try it out. Now, first of all, we want to write down the definitions of the hydrolysis of starch by amylase enzyme. So how can you write that? What you need to do is you just need to copy down the whole sentence here. Hydrolysis of starch by amylase enzyme is, now you start the definition, is the process where amylase breaks down the starch. So the process where amylase break down the starch is the definition of hydrolysis of starch by the amylase enzyme. The second point you need to write down is to determine by, measure by, and shown by. And you can just continue the sentence like this. And it is shown by the time taken for iodine solution to remain yellow. Now, the time taken for the iodine solution to remain yellow, this will be our RV. We are doing this in our experiment. So we're actually measuring the time taken for the iodine solution to remain the yellow. And this is the RV. The last point is you must write down this hydrolysis of starch will affected by what, which is our MV in the experiment. So let's try to write it down. Hydrolysis of starch is affected by. See, I use the words affected by. By what? By our MV. The pH value of the solution. So this pH value of the solution is actually inside the experiment that we do this reaction in different pH value. This is actually our MV. So this formula can be applied in other questions as well. So you just need to go through more questions like this, then you'll be very familiar with the formula here. Alright, so tips number 7. Predictions. Alright, so prediction is one of the very popular question in paper 3. So you need to predict something. What happens if we repeat the experiment with adding something else, something new into the set of the experiment? You need to predict. So for example, like this question, the experiment of the albumin suspension at 15 concentration is repeated by adding 3 drops of concentrated hydrochloric acid to the mixture. So let me brief you a little bit on what is this experiment is doing. So first of all, we have a set of experiment, and this experiment, it has the 15% of the albumin suspension inside it. We put in the pepsin, and the time for the pepsin to digest the 15% of albumin suspension is 5 minutes, let's say. Now we are going to repeat this experiment, but then we are going to add in some other stuff into the experiment. So let's draw it out again. This is the first set, and this is the second set of the experiment. 15%. Also, okay, we add in the pepsin as well. But this time we add in another 3 drops of hydrochloric acid. Now remember, the experiment 2 is something that you need to imagine. You need to predict. You are not going to do it in the lab. You are not going to do it in the lab, but you want to predict it. So if the question asks you to predict, you want to compare the result of the previous set, which is the set 1. Can you see that? So because they are using 15% of suspensions here also using a 15%. That's why you need to compare back to the original set. So let me try to give you the answer here. So predict the outcome of this experiment. Explain your prediction. The time taken for the pepsin to hydrolyze albumin suspensions will shorter than 5 minutes. So you can see I actually write shorter than 5 minutes. you shouldn't give the exact number or the exact time in the questions. For example, like you know it is shorter than 5 minutes, then you just simply tempa 4 minutes. Okay, it will be like 4 minutes for the pepsin to hydrolyze the albumin suspension. This is wrong. You cannot give the exact amount of time because you didn't do the experiment. So what you can do is you need to predict and compare the result with the previous experiment. That's it. You can get the full marks for this question. Alright, we are here to the last tip of paper 3. Study smart. Alright, so well, as we mentioned earlier, you only have 45 minutes to carry out the experiment and do and answer all the questions on the paper. Those experiments which is longer than 45 minutes is definitely not going to come out in your paper 3 exam. So when you are studying the textbook, try to eliminate those experiments which has longer than 45 minutes. So for example, like the effect of presence of phytohormone on the food ripening, we want to study how the phytohormone affect the ripening of the tomatoes, and this might take you few days to do the experiment. Am I right? So this is definitely not coming out in your paper 3. How about the capture, mark, release and recapture technique? Your teacher is not going to ask you to capture some turtle and then wait for another days and come back to check which turtle will have the Indian ink on their abdomen. You are not going to do this, alright? So your teacher is not going to do that as well. So remember, there's no dissection because how many mice you want to kill? If let's say one class we have 45 students, you need to kill 45 mice, you are not going to do that. So no dissections, no capture, mark, release, recapture technique, no which is longer than 45 minutes. Alright? So go study now. Remember, look for those experiments which is less than 30 minutes because you need some time to answer the question on the paper as well. You are not going to spend all the times in just doing the experiment, am I right? So for example, like TCPIP, the vitamin C ascorbic acid experiment, methylene blue, diffusions, osmosis of potato, those kind of experiment which is short and simple, easy to get. The easier it is, the higher chance it will come out in your exam. Alright, next, easy to prepare. Try to imagine if you are a teacher, are you going to prepare something which is very hard to buy it? For example, are you going to buy a vaccine just to study the immunity? No, you are not. So just remember, okay, find some experiment which is easy for your teacher to prepare it. So last one is the safety. The experiment must be very safe because your teacher is not going to send you to the clinic or hospital halfway during the exam. Am I right? Those experiments which includes the strong acid, strong alkaline, maybe boiling the water, those experiments is considered as a bit dangerous, so it's less likely to be coming out in your exam. Alright, so that's all for my 8 tips for biology paper 3 experiment. Hope these 8 tips really help you guys. Please help me to like and subscribe, and I will see you guys in the next video. Bye bye.